ക്രൊമാറ്റോഗ്രാഫിയിലെ ഒരു പ്രധാനപ്പെട്ട ടെക്നിക്കാണ് അഫിനിറ്റി ക്രൊമാറ്റോഗ്രാഫി. അഫിനിറ്റി ക്രൊമാറ്റോഗ്രാഫിയിൽ രണ്ട് മോളിക്യൂൾസ് തമ്മിലുള്ള അഫിനിറ്റി, അതായത് ആൻറ്റിജൻ, ആൻറ്റിബോഡി, RNA, DNA തുടങ്ങിയ മോളിക്യൂൾസ് തമ്മിലുള്ള അഫിനിറ്റിയാണ് സെപ്പറേഷന് വേണ്ടി ഉപയോഗിക്കുന്ന ബേസിക് ഫിനോമിനൻ. എന്താണ് അഫിനിറ്റി ക്രൊമാറ്റോഗ്രാഫി? അതിലുപയോഗിക്കുന്ന രണ്ട് പ്രധാനപ്പെട്ട ടെക്നിക്ക്, ഒന്ന് GST ടാഗിങ്, മറ്റൊന്ന് ഹിസ് ടാഗ് അല്ലെങ്കിൽ ഹിസ്റ്റഡീൻ ടാഗിങ്. ഈ മൂന്ന് കാര്യങ്ങൾ നമുക്ക് ഇന്നത്തെ വീഡിയോയിൽ കാണാം. ആദ്യം എന്താണ് അഫിനിറ്റി ക്രൊമാറ്റോഗ്രാഫി എന്ന് കാണാം. രണ്ടാമത്തെ ഒരു പാർട്ടായിട്ടുള്ളൊരു വീഡിയോയിൽ എന്താണ് GST ടാഗിങ് എന്ന് കാണാം. മൂന്നാമത്തതിൽ എന്താണ് ഹിസ്റ്റഡീൻ ടാഗിങ് എന്ന് കാണാം. ഇത് മൂന്നും കൂടെ ചേർത്തുള്ള വ്യത്യസ്തങ്ങളായ മൂന്ന് വീഡിയോകൾ ചേർത്തുള്ള ഒരു ക്ലാസ്സാണ് ഇന്നത്തേത്.
Let's talk about affinity chromatography. Affinity chromatography is a chromatography technique in which separation occurs based on the specific interaction between the two molecules. This specific interaction can be between enzyme and a substrate, receptor and a ligand, protein and the nucleic acid, etc. Let's see different components of the affinity chromatography. The affinity chromatography is usually carried out in column, which is filled with the supporting material or the matrix. The matrix should have the following properties. It should be chemically inert, should exhibit good flow properties, should have sufficient functional groups for the covalent binding of the ligand. Different materials like agarose, polyacrylamide, polystyrene, cellulose, silica, etc. can be used as a matrix. The matrix is attached with a spacer M, which is usually made up of CH2 group. The presence of spacer prevents the non-specific interaction of the ligand with the matrix itself. Let's see the working of affinity chromatography. When the mixture of different molecules is passed through the affinity column, only the molecule that have specific affinity for the ligand binds the ligand. The other molecules are washed away. Next, to disassociate the molecule of interest from the ligand, a buffer with different ionic strength or pH is passed. This finally releases the molecule of interest from the ligand and the molecule is obtained in the pure form. The molecule of interest can also be detached from the ligand by using a competitive inhibitor. The competitive inhibitor can further be removed by dialysis or by changing the pH of the ionic strength. Let's see different examples of ligand and the molecule of interest that can be purified with affinity chromatography. AMP can be used for the purification of NAD-dependent dehydrogenase and some kinases. ADP can be used for the purification of NADP-dependent dehydrogenases. Calmodulin for the purification of calmodulin-binding enzymes. Avidin can be used for the purification of biotin-containing enzymes. Metals such as nickel can be used for the purification of proteins that have a histidine tag. Lysine can be used for the purification of rRNA.
Many biological molecules recognize other specific biomolecules and bind to them with high affinity as part of their normal function. For example, hormones are recognized by specific receptor proteins. Antibodies recognize their respective antigens. Enzymes recognize their respective substrates. This ability to recognize a partner molecule is harnessed in affinity chromatography. For instance, if we conjugate, that is attach, an enzyme onto the surface of Sephiro's beads, we generate a stationary phase that can isolate the substrate of that enzyme from a complex mixture. One pair of biomolecules that bind to each other with high specificity are the enzyme glutathione S transferase GST, and its substrate, glutathione. This pair is involved with the removal of toxins from the body. Glutathione is a tripeptide with sulfhydryl group shown here in red. The importance of the sulfhydryl group is reflected in the abbreviation we use for glutathione, which is GSH. Glutathione S-transferases are enzymes that seek out toxic, unfamiliar molecules in cells, such as drugs or pollutants. When they find a toxic molecule that is too hydrophobic to be excreted, they recruit glutathione. They attach the glutathione to the toxin via the sulfur group. Glutathione is water-soluble, so the toxin can now be excreted from the cell and from the body. If glutathione molecules are conjugated onto Sephiro's beads in a column, we can isolate glutathione S-transferase from a complex mixture by affinity chromatography. This is more useful than you might expect. Suppose we want to produce large quantities of a protein of interest. Let's call it protein X. We could insert the gene encoding protein X into bacteria. The bacteria will now produce large quantities of protein X. We can now lyse, that is, rupture the bacteria. But how do we isolate protein X from this complex mixture of proteins, DNA, and membranes? To overcome this difficulty, we add the gene for GST to the gene encoding protein X. Now, when the bacteria produce protein X, the protein is fused to the GST protein. This technique is called protein tagging. The GST is acting as a tag to allow easy isolation of the protein of interest. When we lyse the bacteria now, protein X can be isolated by affinity chromatography on a glutathione Sephiro's column. How do we get the protein X GST complex off the column? Remember, it is the GST that is actually bound to the column via glutathione. We wash the column with a solution of high concentration glutathione. How does that work? The high concentration of glutathione displaces the column-bound glutathione at the GST binding sites.
GST ടാഗിങ് പോലെ തന്നെ നിക്കൽ അഫിനിറ്റി ക്രോമാറ്റോഗ്രാഫിയിൽ ഉപയോഗിക്കുന്ന ഒരു പ്രധാനപ്പെട്ട ടെക്നിക്കാണ് ഹിസ്റ്റഡിൻ ടാഗിങ്. എന്താണ് ഹിസ്റ്റഡിൻ ടാഗിങ് എന്ന് നമുക്ക് നോക്കാം.
So to summarize, GST tagged proteins can be easily isolated using a glutathione GSH column. The GST tagged protein is trapped on the column because GST binds to GSH. The GST protein complex can be removed from the column by washing the column with a high concentration of GSH. Subsequently, GST can be cleaved from the protein of interest.
In this video, we're going to discuss the principles of nickel affinity chromatography. So as we know, chromatography in biochemistry is a technique that's used to separate all sorts of kind of molecules. This is going to be a technique that's going to be used to separate proteins, but this kind of chromatography is very good for separating a specific kind of engineered protein, which is very, very common. And that's what's called a his-tagged protein. So what is a his tag? A his tag is a segment of usually about 6 histidine residues. So histidine is an amino acid, and you can put 6 of them in a row on usually the N-terminal end or the C-terminal end of a protein. And the histidine tag, so 6 histidines, impart some important properties to the protein. So I'm going to briefly go over to this slide right here. And what you see here is here's my protein, and you see here 6 histidines. They're on opposite sides here, but there's 6 histidines right there. This is my 6x histidine tag, so there's 6 of them. And what these histidines do, based on their imidazole group, which is their R group, they can actually bind certain things. And it turns out that the histidines are really good at binding nickel. And we're going to exploit that property in nickel affinity chromatography, which is going to allow us to separate and isolate proteins with a 6x his tag, which will be added via this process here. All right, so let's talk about how you actually get a 6x his tag or just a his tag onto a protein. It's actually a pretty complicated process. In most biochemistry labs, you would not, at least course labs, you would not actually do this process. It would probably already be done. What you generally have to do is you actually have to isolate the gene of interest. We're going to use what we've done in previous examples in this playlist, the cytochrome C gene. This is the CYCS gene. This is actually the DNA segment of it. Okay, it's in green. I also have to have what's called an expression vector. They're a lot more complicated than this. These are circular plasmids that are typically found in bacteria, but some of these expression vectors can be used to force a cell to express a certain protein. So right here I have what's called an insert site. This is the site where I'm going to basically break the plasmid apart and insert this gene for cytochrome C. On the left of the insert site, I usually have a couple of things. One, I could have the 6S his tag. Being on this side, this would actually add the his tag on the N-terminal side of the protein. Sometimes you'll see something on the right side. It could be a fluorescent tag, like green fluorescent protein. In one of the labs at the school I used to go to as an undergrad, they actually have a setup very similar to this. So I have on the N-terminal side, or 5' side, because it's DNA, I got the his tag. On the 3' end, which will be the C-terminal side of the protein, I have a GFP tag. So through a process that's more complicated than this, you're going to split open this plasmid, you're going to insert this segment. This is the gene for cytochrome C, and notice how it inserts right here. I'll have the 6S his tag, the cytochrome C gene, and then the green fluorescent protein tag. You close this plasmid, so now it's closed, and you can actually force this into certain bacteria, such as E. coli. That's called transformation, and this is a vector because it's going to transmit this gene. You're going to force this vector into cells like E. coli, grow the E. coli, and then the E. coli will be forced to express this protein for cytochrome C. However, on the N-terminal side, now it's going to have a histidine tag, so 6 histidine residues right at the N-terminus, and it will also have green fluorescent protein on the C-terminal side. This green fluorescent protein is not necessary for nickel affinity chromatography, but it can help tremendously in downstream steps later on in your procedures down the line. So this E. coli will now express this protein. Now, if I want to actually isolate cytochrome C, I can use this histidine tag in nickel affinity chromatography to my advantage. Let's go to the next slide. So first of all, I'd have to take these bacteria, which in this case are E. coli, lyse them, and that will release this protein of interest. So I now have all this protein right here. This is my cytochrome C with the GFP and his tag, right? Then I'm going to incubate this sample with what's called nickel NTA agarose beads. So nickel NTA, that's what these are shown right here. It's nitridotriacetate. That's what NTA is. You have a resin bead right here that's connected to this nitrogen that has 3 acetates coming off of it. So these acetates are negatively charged, and these 3 acetates together will actually bind this nickel cation. Okay, the nickel is charged. The acetates are all negatively charged, and they will chelate or hold that nickel in place. Okay, I've got one of those NTAs right here, another one down here, and they're both bound to nickel cation. So when I incubate the protein with the nickel NT-agarose beads, what happens is the histidine residues of any protein with a 6s his tag, those histidines will also bind to the nickel. And so what will happen is, on one side, the nickel is bound to the NTA, but on the other side, it's bound to this his tag. And so that's one way of holding on to this protein with the his tag. Generally, once you've incubated and you have now bound protein, that is, the his tag is bound to the NTAs, you usually wash it with a salt solution. So any proteins that are not bound to the nickel will just wash through and you're done with those, basically. Okay. This interaction between the histidines and the nickel is very strong, so it's generally not enough to, this salt is not enough to wash out this protein. But then once you've gotten rid of pretty much all the proteins you're not interested in, then you're going to elute this protein by throwing in what's called a competitor. This is just straight imidazole. It's not an amino acid. This is just the R group by itself, the molecule imidazole. And what the imidazole will do is it will compete for the nickel because it's the same functional group as what's on the histidine R group. So the imidazole will compete for the nickel, and it will essentially knock off or displace the his tag, and then the protein with the his tag will fall off, and it'll allow you to elute those, and then this fraction right here should pretty much just contain polyhistidines, which is the 6s his tag. And so now you've isolated your protein of interest into this tube, which in this case is our cytochrome C, just our example here. So now I have two fractions basically. I have the one that has the polyhis tag, which is my cytochrome C, my protein of interest. Over here I have a bunch of other proteins. These are the proteins that didn't have the his tag and therefore washed out with the salt solution. So if I compare these two samples, let's talk about before nickel affinity. So basically this could be my, my wash step right here, the sample from that, or before I ever did the process, I could test that through an SDS page and I'd still see all of these proteins here. Okay, because I haven't yet separated out the proteins that don't have the his tag, so I have everything. But if I just test the fraction that should only have the his tag and therefore only my protein of interest, I might just see this, okay, in this lane. Most everything else has been filtered out, and so the only thing really remaining is my protein of interest, which had the his tag. And so what you can see here is in the nickel affinity trial, so after I actually get this fraction right here, it's much purer because all this other gunk down here, all these other proteins I don't care about, are gone. And so that's the principle of nickel affinity chromatography. Now, this type of chromatography is a type of affinity chromatography which is widely used, but this type is really only applicable when you have some entity that will bind to the nickel. And really the most effective thing that binds to it are these his tags. And since his tags are really common in genetic expression, for forcing bacteria and things to express, it's a very common technique to separate those proteins because this is the most effective group just about to bind to the nickel. All right, so again, a purification/separation technique, but very useful in molecular biology and biochemistry. All right, so hopefully the principle of nickel affinity chromatography makes sense to you, how you actually get the protein to be expressed with the his tag and the GFP. Again, this process right here is actually extremely complicated. I just showed you the very basic idea, and then hopefully it makes sense the actual general process of the nickel affinity chromatography. Again, it's a longer process, but this will get you the basic idea.